got a great set of attendees today. Really looking forward to this discussion. So I'm Patrick, and I'm excited to be here with Dr. Mark Kotter, who's the CEO of Bit.Bio. Focus today is really on precision medicine research in MS and what's on the horizon. So agenda today, I'm going to give a quick introduction to myself and Sano, and just a little bit around some unanswered questions in genomics from a precision medicine perspective. And then Mark's going to talk about himself, Bit.Bio, and the amazing work they're doing around cellular reprogramming and cell therapies. And then we'll have time for Q&A and discussion. So we are recording this. If you don't want to be recorded for any reason, then please just let us know. You should be able to add questions at any time through the chat. You can raise your hand in the chat if you have a question in real time. And if you have any issues, just let either Claire or Ross know. So Sano is a platform for patient-centric precision medicine research. So we have an online platform that anyone can join, and we help match people with research studies and help them learn more about themselves through personalized reports and other content. We really feel like there's an opportunity to improve the participant experience in research, and that ultimately bringing patients and researchers more closely together will help us accelerate research. And we do at-home testing for whole genome sequencing and other kinds of omics to help make participating in precision medicine research just a lot more convenient and democratized, so everyone has access to this. If you're interested in learning more, you can obviously visit our website. I just wanted to start off with a little bit of a framing from my perspective of what personalized or precision medicine actually means. And in my view, I think the terms are really interchangeable. Some people like one more than the other. But ultimately, it comes down to the four Ps: prediction and prevention, more precise diagnoses, more personalized and targeted interventions, and then a more participatory role for patients. That fourth one, I think, is often overlooked, and it's a really important one to us as a company. We think that there's a huge importance to actually involving patients or participants in both the research process and also more closely in healthcare delivery, because often you are the expert on whatever it is that's affecting you, and you're the person who can determine what your priorities are. So just as an example of kind of participation in action, we recently did a survey with around 1,500 users of the Sano platform with MS, and you can get a really good picture of the, just the differences in people's experiences and where they are in their journey. So some people have been diagnosed for 10 or more years. Some people, more recently. There's also, as we know, a number of different subtypes of MS, and every 5 or 10 years, there's a kind of rethinking or re-understanding of exactly what the disease is. And so this is kind of an evolving set of labels as well. And then finally, we also know that people experience MS really differently. So around 92% of users of Sano experience fatigue, around 50% experience vision problems, for example. And we're really only beginning to scratch the surface as to why any one person experiences this disease and others in the way that they do. So asking participants about their experience and then combining this information with genomic and medical information, we think is a really powerful tool to ultimately make research and treatment more personalized. So just as a quick overview of where we are with respect to genomics and MS, which is our focus, and my expertise is in genomics, although I'm certainly not specifically an MS expert. There's been a number of really large scale genome-wide association studies that are looking for relationships between rare or common genetic variants and developing MS. But there's still a number of unanswered questions. So common ones that come from patients and researchers alike: will my family members develop MS? What does the future look like for me? What treatments or lifestyle modifications might work best for me? And also importantly, what differentiates people who are going to have a more progressive form of MS from people who may not progress as quickly. And really interested to hear from Mark because they're working at the forefront in some of the early stage research that could ultimately hopefully lead to more treatment that's focused on remyelination or repair, which is obviously a really exciting area of research. So I'd close out here, and I'd just like to say we're at the early stages of starting an at-home testing program to power research in MS. So I'd invite you to get in touch with Claire, who's on the call, Claire at sanogenetics.com. If you're interested in learning more about this, we're a very open and collaborative company, and we'd like to work with patients, patient organizations, healthcare providers, and academic and industry researchers who are interested in this. What we'd really, our vision for the future is that not just in MS, but in genomic conditions in general, that everyone should have access to genetic testing, full control and transparency around how their data is being used, and be able ultimately to power personalized medicine research and healthcare in these conditions. So please do get in touch with us if you're interested in learning more about the work we're doing here. I know there are a couple of people on the call who Claire's already in touch with, and that we've already started working with on this. So I'm going to hand over to Dr. Mark Kotter here, who's the CEO and founder of Bit.Bio. They have an amazing platform technology for creating precision engineered human cells. And Mark has done, I think, did you do your PhD or postdoc work focused in MS? Is that right, Mark?
Yeah, correct. I had the pleasure to work with Robin Franklin in the early days on CNS remyelination. And it continues to be an important theme of my academic work, and is that ultimately the reason why we have developed our platform. Patrick, very happy to share slides, but I think I need to probably... you doing it for me?
Yeah, I can do it for you if you'd, and then you can just tell me to advance if that works.
Right. Okay.
Unless you want to share. Do you prefer to share?
I think it's probably easy. Yeah, I feel a bit more comfortable if you don't mind.
I will let you share. You should be able to share now.
There you go. So I just wanted to sort of spend a little bit of time, very short, on Bit.Bio. It's a synthetic biology company that is using a reprogramming paradigm, a coding paradigm for health issues, and really, I think what this opens up is the idea of precision engineering of human cells, cells are a bottleneck in a wide area of medicine, from basic research to obviously drug discovery, all the way to cell therapy. And we already see quite a lot of interest in all three areas. So the fundamental problem that we, I think, have solved are these three things that are the bottleneck for any of these applications. Really, precision of generating a cell from a stem cell, so mixed cultures, subtypes of cells, the consistency by which you can achieve this, and the scale by which you can achieve this. And all of them are important if you think beyond, single cell or few cell experiments, if you want to go into more systematic studies. And I'm going to take you away from MS. This is a best-in-class differentiation protocol that I just picked out of the literature, and because it's quite visible. You can see a muscle cell differentiation protocol, 40 days, lots of steps, and the purity is sort of restricted, let's say. Is that it also highlights the other big important bottleneck in stem cell differentiation, which is the immaturity of the cell. So people get very excited when they see a muscle cell, a muscle fiber with multiple nuclei. So what we have done is we have taken this away, and I'm just showing you here pluripotent stem cells that are reprogrammed into neurons. And you can see that the entire culture turns into neurons. And then after 4 days, they start to just create more and more connectivity and become and form a neural network, a real neural network, essentially. And after in terms of time to specification, is 4 days. Interestingly, electrical activity emerges around 10 days. And you can see it on the video. It's very, consistent. Very exciting as well. These networks start to function. That means a good readout of that would be synchronized network activity after 20 days. And that really compares to about a 100-day expedition, so 100 days faster than traditional approaches. And this is our external validation. Charles River have tested this product and other cell products that we're producing for its utility for high throughput screening. And how is this possible? It's based on 3 axioms that have emerged over the last, since 1987. So gene regulatory networks encode cell identity. And these, again, are controlled by transcription factors. So if you activate master transcription factors, you can evoke a new cell identity. And the first of this happened in the 1980s, a muscle cell. That's why I get caught back to the muscles. And then, of course, Yamanaka, complete breakthrough in terms of iPSCs. And since then, the field has, expanded. Our addition, so in detail, that means we can skip those intermediate stages and go directly from a pluripotent stem cell to a neuron. And this solves, this has the ability to solve the inconsistency. The inconsistency in reprogramming, that's another thing that required solving. And this is some data that actually haven't published, which basically shows that the inconsistency is related to the inefficiency of turning on that program, the control of transcription factors. And why is that the case? Because pluripotent stem cells are very good at shutting down external transgenes. And you can see this. This is a cassette with a nuclear tag. After 2 days, every cell seems to be red. And then after that, extremely inefficient. Every red nucleus, nearly every red nucleus has gone away. And the very few cells, 1 in a million approximately, that had a nucleus still have reached the oligodendrocytes lineage stage. So really what we've created is a system that allows you to switch on and switch off genes in human stem cells, and it's based on a gene targeting strategy. And it leverages the protection of so-called genetic safe harbors. And this has put us into a different league. So now we can produce cells that are beyond the high throughput screening threshold. You need very consistent cells for large-scale experimentation. And not only that, our customers can, get these cells and put them into culture, and within 2 days start their screening experiment. So we really very much simplified the approach. And in terms of purity, this particular product is 99.999% pure. Scale, yes, you can have as much as you want in terms of the cells, and they are quite unique in terms of function. We discussed the MEA properties of the cells. I want to just dwell on that concept of hierarchical transcription factor networks and how they specify cell identity, and how we use this to precisely engineer cells. So this is a little bit of a scientific deep dive before I hand over. So this is really an experiment that looked at the program that we just saw in the video, stem cell to neuron. And we used an early stage, obviously iPSCs, the definitive neuron stage, day 4, and some later time points where we see electrical activity and synchronized electrical activity. And it's a quite complete data set. Again, I haven't published that yet. And so all we wanted to understand is, how does it work? I mean, what's under the bonnet, essentially? And here you can see some ataxic data. These are nucleosome positioning. So you can basically see the activity of sites, the opening of sites that are under an NGN2 promoter, and a promoter that NGN2 recognizes. And so what you see very quickly, there's a lot of activity in the NGN2 genes, downstream genes, and after 4 days we can switch it off and the activity dies down. And what this does is it basically takes the stem cell population, here this is a single cell sequencing experiment, directly into what I would call a day 4 baby neuron. So a bona fide neuron that can't do anything else as immature. And the important thing is, it's a very direct path. And we've seen this on the video. And the reason why this is possible is because of the precision by which you can control these gene regulatory networks. So this is a bulk-seq experiment. By the way, all data that Kaiser did in his PhD in my lab, that has 3 biological replicates. In terms of, first of all, looks impressive, the overlap. I've not seen this in other cell systems so far. And it's a bit like clockwork. And that's probably, leading to what we again saw in the video, that synchronized, reprogramming of the cells. I initially thought this is going to be a completely synthetic process, but we looked deeper and did a timeline experiment, and this is bulk-seq data, and you can see that actually the starting pluripotent stem cell, they whiz through a neuroprecursor stage. Now, I'm not saying this is a typical neurostem cell, and that we haven't tried to capture that stage, but it is very highly reminiscent of an intermediate. So what I'm arguing here, at least there's a partial recapitulation of development, a concept that I always sort of tried to fight, but actually the data tells me that no point fighting nature. And the other thing is, at that point, at day 4 to day 21, of course, we see significant changes, but these are mainly related to maturity markers. Neuronal differentiation, synaptic transmissions are the main driver of the PCA. Now, that is something that I'm really sort of also very interested in understanding better. So here we're looking at ATAC-seq data. That's the sequencing method that looks at open versus closed chromatin sites. And again, we can see a very nice overlap of the 3 independent biological repeats. And then looking at the data in more depth, what we observed was that there's a massive rearrangement of the chromatin happening in the first 6 to 12 hours. So you can see a huge amount of sites that open up, that obviously are driven by the NGN2 that we activated. And then after that, it becomes a much more linear process, and which plateaus. And the opposite, more or less, is true for closing sites. You see very little closure at the early stages, and then you can see how the sites close off as time goes on during maturation. And I think that data gives us a glimpse of what cell identity actually is, and how the epigenetic sort of system plays into the concept of cell identity. So one way, and this is a wild hypothesis, so it's an inspiration that I've drawn from this graph, is that probably the loss of activity, so the closure of chromatocytes, may be a function of an energy-preserving mechanism. Sort of you collapse the DNA that you no longer require. And I'm not saying that this might not add stability to the cells, but what I can say that the cells here are equally stable. You can't get them back into any other cell state than a neuron state before that closure has occurred. So this is not MS, but it's a monogenic. So actually again a polygenic condition, and just happened to be the first one that we modeled. And you can see, you see condition phenotypes. So here, they didn't manifest in terms of changes of connectivity, but there was frequent increased frequency in the patient. No client-derived lines with regards to basal electrophysiology, so activity, and then reduced synchronized firing pattern. And of course that takes us to a different level. It was just a proof point. But this is something I'm really very excited because people say, yeah, okay, you've now got a cell type and it's fine, but actually cells don't always act by themselves, and sometimes the interaction of cells is important. And the problem, of course, is how do you model this in a human system? And one of the proposed solutions is organoids. And as much as I love organoids in terms of sort of basic research, they're quite impractical. Often 6 months in culture. I don't know whether you've seen organoids. Every organoid looks completely different from the next. I mean, macroscopically, extremely different. Now, that's not very conducive, again, to certain experimental approaches. And here's the alternative. You print cells in a 3D matrix, individual cells or individual cell groups. You can arrange them in 3D exactly how you want them. You can scale this, as you wish. And then you can observe, migratory behavior. So they printed our neurons and astrocytes in the inside or the outside of the, we call them now printoids. And you can see that astrocytes don't like to migrate versus neurons, baby neurons do like to migrate. And that allows you to sort of even spatially control, your systems. Now, this is something we're working on. This is not a product, but it's very exciting. This is a product. So you can get these cells, you can take them out of the fridge and basically start experimentation. Literally on the first day, you're going to see process outgrowth, and within days you can see connectivity. And this is much more sophisticated data that Charles River has produced in terms of the MEA reading. Working very hard to make more cell types available, and part of them for MS, very important, are the oligodendrocytes, obviously. We are also very interested in immune cell space, microglia, very important, etc. So this really creates a new toolbox for studying MS. And with this, I'd just like to say a quick thanks for giving me the time to present that.
All right, great. Thank you so much, Mark. For anyone who's interested in asking a question, feel free to use the Q&A box on the bottom. I have a couple. You have time. I have a couple that I'm going to ask Mark, but please do feel free to put anything in the chat. The first one, Mark, that I wanted to ask was if you could kind of paint us. I know this isn't ready for cell therapy applications yet, but I wonder if you could kind of paint us a roadmap of what needs to happen before you could conceivably start to engineer cells that could be used in repair and neurodegeneration or other kind of CNS applications.
So I think you probably picked a hard group of conditions, but we do want to get into the clinic within the next 3-4 years, and maybe we will tier it so that CNS is not the first, but we'll sort of see what the timelines are. I think we're already in a position where the cells have demonstrated superiority in terms of preclinical large-scale applications. And that makes us very confident that will translate into an advantage in the clinic as well. Obviously, we need to work through, the GMP process, the regulatory processes, and have initiated those programs. What are the hurdles? So, because there are not so many stem cell, well, there's no stem cell product out there, and there are very few trials. I think regulators are also working and learning and defining the regulatory landscape, and that might be a little bit of a that can cause issues because you essentially have to divvy out what you need to prove in order to show that your product is safe. Other bottlenecks. So we have much fewer bottlenecks with regards to, our differentiation or reprogramming protocols, because we don't need serum or anything like that. It's all fully defined. So the transition to GMP is probably much, much easier than other paradigms. Developing the right QC criteria. I mean, we're going to the nitty-gritties here, but no, we're quite excited about the opportunity to take this into the clinic.
Yeah. So it's a very simple question, I think, but is it? Would you be reprogramming an individual's cells, or would you be, having a kind of general stem cell population that you then kind of transplant into others? Which of the, or is it something else entirely?
No, I want to highlight I'm a scientist and not a politician, but the answer is it depends. And so, look, there are conditions where off-the-shelf products that are not matched are an advantage. If you think about over-encapsulation technologies, and actually the mismatch is a safety network. And then you have indications where you need a certain level of matching. And you could use, and you can use sort of the principles of organ matching. There's a lot of experience around how you best, match donor and recipient. And of course, here CNS conditions are the big advantage because of the protective environment of the CNS. We feel that an off-the-shelf approach in the CNS is very attractive. And then, is there a route or rationale for patient-specific derived cells? Yes, there's also a huge cost factor attached to it at the moment. What would be the ultimate solution? I think there's two ultimate solutions: a cell that you can transplant into everyone. And that is the concept of universal cells or hyperimmune cells. And I think the field is making progress there. And the other, maybe even better option is a process that's so slick that you can use patient-derived cells. And again, I think, it's not only us that are working to pushing those boundaries, but fundamentally these are the two routes for the future.
Yeah, no, that helps. That really clears it up. Thanks. And in terms of MS in particular, which I know you spent a lot of your early research career on at Bit.Bio, you have a very wide lens, I think, in terms of what your technology can actually do. I wonder if you could talk a little bit about how the field has changed since you were working on that, and maybe a little bit about what you see the future being.
So, I must recognize I'm not an MS researcher. I'm very interested in the regenerative response, and in particular the biology of the oligodendrocyte. And with all these preambles, I think we've learned a lot about MS. Even you showed it. There's very robust evidence now that it's a condition that's driven by the immune response, by the immune system, which essentially means in order to model it, to understand it, you need to understand the immune system and how it interacts with the central nervous system. So causality is not an area that I'm very familiar with. And as you say, there is we're going through cycles in MS research in which, certain ideas gain more prominence than others. And I wouldn't be able to commit personally to knowing or, backing more than, that the immune system is a very important driver. And we know that from, the antibody treatments that are in place. And then in terms of the regenerative space, there's always this dichotomy, releasing the regenerative potential of the endogenous OPC population, using, for example, small molecules or other approaches. And the second possibility is, of course, transplant. And here, again, I think you've got to go precision. You need to know what would be best for what. And I can speculate here, but I think we're just entering that new wave.
Absolutely. We actually have a question through the Q&A that maybe would be good, that someone was wondering, is the oligodendrocyte and microglia technology in process or currently available? What are you able to offer in terms of those two cell types?
So we're working towards the beta release of the microglia and the oligodendrocytes. But I don't think my commercial team would want me to commit on any timelines.
So would it, with the kind of following up on the point you had about the CNS printoids, is there a near future where you can kind of model the interaction between all the different important pieces of the puzzle in MS in a kind of model system like that, and get closer to what's actually happening in real biology? Is that the kind of near-term goal for you all?
Let's put it at if near-term goal is next year, then it's not a near-term goal to sort of. But I think recognize how important this is, and the reason why I've showed this is really because we believe very much in sort of integrating different human cell types in a sort of controlled 3D environment. I think we know that brain cells look different when you put them into a 3D matrix. We know that they function probably slightly different. OPCs are, for example, extremely mechanosensitive. So stiffness of the niche of the extracellular matrix has a massive impact on the function. We believe, and that's something that we haven't shared yet, that human oligodendrocytes are even more sensitive to the environment than rodent cells. So lots of reasons why we need to work on these model systems, but these are. That's the next generation of model systems.
Great. So Amanda has put a question to the chat, and two questions actually. I'll start with one that is probably specific for you, Mark, which is, what's the potential extent for repair for people living with MS today? And I know, caveats aside, and is there potential to significantly reverse damage for those who've been living with MS for many years, and if it is how, what do you think is the time horizon for some of these kind of potentially life-changing and game-changing therapies in MS or otherwise?
I think again one has to take a precision medicine sort of view, and you need to consider the stage of MS and the stage in particular of the lesion that you're targeting. So MS, multiple sclerosis, alludes to the fact that all lesions are sclerotic, which means very firm. And those very, very old lesions have reached a stage which is going to be very difficult to resuscitate. But then the sort of more immature sort of MS lesion stages, the subacute group, and of course, especially those partially demyelinated lesions, which are very frequent, and there's a time element in this. Are much more conducive, I would say, to regeneration. And then if you look at the sort of cellular component, you've got axons and neurons and oligodendrocytes, but we know other cell types also play a role. Now, if your symptoms are more the consequence of demyelination, that is probably likely to be a much easier fix than when the symptoms are driven by loss of axons and neurons. And so I think the focus for any trial in MS would be to identify a group of the exact subgroup or a group or the exact subgroups of individuals that have these subacute lesions that cannot be repaired without external, supply of external cells. And you also want to target this to areas in the CNS where you see a big difference. So for example, and I don't want to, we don't have a trial design for this yet, but things where you would notice a big difference is, of course, vision, optic nerve, spinal cord, big difference, certain brain stem tracts. But you've got to be very careful. You need to select the right risk-benefit profile, etc. So this is complex. MS is extremely complex. I think we are certainly getting much better and much closer to clinical translation. But this theme, the MS theme, is a complex and complex issue.
Is it fair to say that the complexity of the disease from a genetic or molecular perspective kind of impacts the complexity or challenge around the regenerative approach that you all take? Do you think the first diseases that you'll see this being applied in will be single gene or more monogenic or molecularly simple, for lack of a better term, or is it can you actually, because I guess when you have something that is a complex disease that is many genetic factors and environmental factors that's taking place over many years, it seems it's almost hard to believe that you could come in with a single fix, but yet we see it all the time in medicine, whereas with a single genetic change, my brain thinks maybe it's simpler to come in with a single fix. But I don't know if does that logic break down?
I think it's a very good concept. I just want to reflect on the fact that we're not dealing with we're not fixing genes here. A cell therapy essentially would be introducing an intelligent drug, so to speak, something that knows what it does into an environment of damage. And I believe strongly, based on decades of preclinical work using rodent models, that if you stick the right oligodendrocyte precursor into a lesion that contains axons that are demyelinated, the oligodendrocyte precursor will not care how the damage has occurred. It's just going to fix the damage. So sometimes complexity doesn't translate into all treatment modalities, but certainly, it has taken a lot of time to come up with much better MS treatments, and we talked about the complexity of actually deciding what to treat and how to treat, endogenous versus endogenous. I mean, again, that needs a lot of thought, and I would never. I wouldn't imagine that I'm in a position to, provide an answer alone. I think that needs.
Great. So we've had a couple more questions come through. So the second one from Amanda asks, would love to hear how Bit.Bio and Sano are working together, and more about our Sano's philosophy on data sharing with patients. And I'll take those in reverse order. So our philosophy is that participants have complete control and transparency around their data. So, it's not ours, it's theirs. It's kind of that simple. And we have a software platform that reflects that, so people have full control over that data. In terms of how we're working with Bit.Bio, we actually aren't working together yet. I'd say we've been admiring the Bit.Bio team's work from afar. We're both based in Cambridge, so Mark and I have known each other for a long time, and they're doing amazing work. We have spoken before about whether it might be possible to collaborate in some way to create cellular models of individual patients with specific genetic or other profiles. And I think it's fair to say it's definitely something that's a possibility for the future. But we haven't actually kicked anything off yet. But we knew that we wanted to have a chance to talk to Mark and learn about what they were up to, because we are working in very similar areas.
No, I'm still extremely excited about the possibility to combine deep genetic profiling with deep clinical phenotyping, and then distilling this into a cell model that is, again, precise and reflective of a particular individual or a group of a subgroup. And if you think about this, what is the best possible approach to studying, any condition? It's really taking that stance, massive amount of data, characterization, very detailed description of the genetic phenotype and the clinical phenotype, and then boiling it down to sort of cell models that are robust and scalable, and you can use them for large scale experimentation. So whole genome CRISPR screens or focused CRISPR screens, I think is something that we're starting to offer now at Bit.Bio. So we have a service or a partnership arm that can do CRISPR screens. And of course, if we were to crystallize this collaboration, I think it could be a real game changer. Quite frankly, we weren't big enough when we started talking. I think we are sort of now approaching 100, and so I think there should be a bit of capacity.
Just to follow on that CRISPR screen, because we've talked a lot about the therapeutic application, but actually the thing that is just as amazing that you all are offering today is the kind of high throughput screening, and I think for me there's a exciting near-term future where you have, as you suggested before, a cell model that represents a single individual or a group of individuals, to the extent that they have something in common, and you can test hundreds of thousands or millions of compounds or gene editing approaches within that specific subgroup. So how does that work with the high throughput CRISPR screening, for example? What are you looking for, and what are you able to do? Drug
screening, just divert very quickly. We currently don't do this in-house. Big pharma do this themselves with CROs that we work with who provide this service. But next year we're going to launch CRISPR screen, very robust and validated platform that we've built, a dedicated team that leverages the unique CRISPR screening approach. I can become very technical here. It's a crop-seq CRISPR screen, which enables you to do pooled experimentation with single-cell readout. And that, really pushes beyond, the boundaries of array screens. It allows you to do, pool even combinatorial screens. We can do activation and knockout or inhibition screens. We're working towards sort of even more complex paradigms. This is all based on our cell models, and it's not a service for other cells. So we'll be very focused on what we have to offer.
Great. I have a couple questions here. So Lean asks, Sano seems to be in a strategic position to centralize different data points from patients. You're already collecting genetic information. Very exciting. Are you looking at integrating patient radiological, immunological, clinical, pathological, cellular profiles? Yes, and it's definitely going to be a hard and a long time before we're able to do all those. But where we'd like to be in the future is that a patient can have as much of this information accessible in one place that they have full control over, and then they can offer access to researchers and healthcare professionals and otherwise if they'd like. We're taking it step by step. So we started with patient generated data through questionnaires, at-home genomic testing. We're looking at integrating clinical information in the near future in particular, as well as immunological. I think one of the challenges in the field is that all of your data or my data is spread out across the ecosystem. So it's really actually hard to gather it all into a single place. But I think as our digital and IT infrastructures start to get better, then this will become more and more possible. And as Mark said earlier, I think when you have all of a huge amount of rich data available, and some of these exciting cell modeling solutions, then you start to be able to, I think, develop much more precise therapies. But it's unfortunately the system is not quite set up for this kind of big data focused research yet at the moment, although it is changing.
I think it's going to be super powerful to pull this data together. And I think what you're doing is taking a bottom-up approach as well as sort of potentially in the future top-down. And I think individuals that have certain conditions that you can reach can be educated to provide this information. And so I'm really thinking about an infrastructure that where you entice individuals to be proactive and collect information really from them directly, in addition to potentially of course radiologically sort of that requires a different infrastructure. But I think this is very exciting. If one brings all of that together, it's going to be a game changer for medicine.
We have another question from Joe. He says, my brother has MS. I have primary progressive MS. DNA shows 17 plus genes involved. How can I participate with Bit.Bio? Do you, Mark, have a patient, do you work with patient organizations or individual patients? I know you all are kind of spending a lot of time in the lab developing the technology, but probably do have a lot of relationships with patient organizations who are doing a lot of important research in these areas.
So quite frankly, no. We're a cell engineering company at Bit.Bio, and we are obviously connecting to sort of users, but we would be very happy to interface with patient organizations. But I think that would be taking us out at the moment into a territory where you are much better placed. And I think that's really where collaboration comes in. And I'm a very strong believer in collaboration, because Bit.Bio can't do what Sano Genetics does, and quite frankly shouldn't do it, because I've got a team of cell engineers, stem cell biologists, clinical experts in cell therapy, and that is quite different from the team that you have generated. I think where they potentially start touching is at the clinical translation side, but even there, a company that is focused on therapy will be focused on a particular, getting this done with as much possible buy-in from individuals that are potentially being treated, but doesn't have the breadth that Sano Genetics would ever have, because of course the breadth is your value proposition.
Yeah, I think it's a key point that we can't do everything, and that's the value of these collaborations. Well, I think we're running up against time here. If anyone has a last minute question, then feel free to put it through the Q&A. But otherwise, I'd just like to say thanks so much, Mark, for taking the time to be part of this, putting together the presentation, and continued success on your great work at Bit.Bio. Hopefully we can do a 5-year anniversary of this and have quite a few of these things headed to the clinic.
And a lot of collaboration on our side. Patrick, thank you very much for inviting me and also keeping the companies in touch and I'm a clinician at heart. I love being asked clinical questions, and my focus has always been the clinic. Thank you for giving me the opportunity to sort of also present our work here.
Yeah, I forgot to mention that in the beginning. You are spending many hours a day seeing patients, right? This is you haven't stopped your clinical job.
So I have a clinical academic job, which essentially gives me a lot more flexibility. But I'm at heart a clinician. And I think it adds a unique perspective. If you have to stand in front of individuals that suffer from a condition that you are treating, it provides a completely different perspective on life, on the work that you're doing, and I feel extremely privileged that I am able to uphold this. So I hope I will be able to continue.
Great. Well, and just to answer the final question that Joe put through the chat, we will. This has been recorded, and we'll share it with everyone afterwards. So we'll put it on YouTube and on our website, and send it to you all, so you can feel free to refer back or share it with others. Well, thanks so much, Mark, and enjoy the rest of your day, and we'll catch up with you soon.
Thank you.
Thank you.